Hello and welcome dear viewers to the video on stability data evaluation and shelf life estimation. In this video we are going to see how the stability data generated after the stability studies is evaluated and how the shelf life or expiry period is assigned to the formulations based on that stability data. So there are some scientific considerations for giving the shelf life period and the expiry period to the pharmaceutical formulations. The guidelines dealing with the stability studies are ICH guidelines Q1A to Q1E. Also, the US FDA guidances are there and EMEA guidelines are there for stability estimation. Shelf life or expiry period is the time period or is the period from manufacturing till the product meets the predetermined specifications of quality for safety and efficacy. In simple way, the shelf life or expiry period is the time period in which the product is safe for consumption and it is with the efficacy as per the labeled indication. The manufacturers of the pharmaceutical formulation guarantees the product will meet all the quality parameters during this period. Generally, the expiry period or shelf life period of 2 years, that is 24 months to 36 months, that is 3 years, can be assigned for the formulations. On the other hand, there is a retest period and the expiry period for drug substances or active pharmaceutical ingredients, that is API. Generally, APIs have higher or longer expiry period compared to formulations because the formulation contain the number of components and undergo the processing, but the APIs are fewer and that's why the APIs may have higher or longer expiry period compared to the formulations. So formulations are safe for use till the expiry period, and expiry period and shelf life is determined from the stability studies. The stability studies are performed on the basis of ICH guidelines and the scientific principles. Also, as per the research and development data for storage, these formal stability studies are performed. The aim of stability studies is to establish the shelf life and expiry period, to establish the label storage conditions or instructions, and also to establish and support the packaging system or container closure system. After stability studies, the shelf life period is assigned and the storage recommendations are provided. Also, the packaging system is finalized. For example, if you are developing a tablet formulation and you are performing a stability study in number of containers like SDP container, blister with material of construction or MOC of alu, PVC and PVDC, or SDP bottle with desiccant, SDP bottle with molecular sieve. And after completion of the stability studies, you come to know that the product is stable in alu and SDP with desiccant. So, as a generic applicant or as a manufacturer of the formulation, the packaging system is finalized for marketing. The stability study data is required in detail for the storage recommendations and the packaging systems. The specifications for CQAs are evaluated in the stability studies. CQAs are nothing but the critical quality attributes like assay, dissolution, microbial attributes, and the organic impurities or residual or the related substances. The effect of the storage conditions is evaluated in the stability studies, and the effect of temperature, humidity in the storage period is evaluated. Also, the effect of formulation aging is studied. So this is the aim of stability study. Now we will see the CQAs which are going to be evaluated into the stability studies. And consider a oral solid formulation, maybe tablet capsule or soft gelatin capsule. The CQAs are assay, that is potency, related substances or organic impurities, dissolution, because dissolution is a performance test for tablet formulation and capsule formulation, and microbial testing. Other tests are also done on the stability samples during the stability intervals and after the completion of the stability period. So those tests are water content, disintegration test or disintegration time, hardness test, and any other impurity which the applicant want to study during the stability period. So these are the CQAs which are evaluated in the stability studies. Now the stability data of 3 batches is required. As per the regulatory requirements, 3 batches are required. These batches are known as pivotal batches or exhibit batches, and all these batches are to be stored in the stability studies in the intended marketed packs. The stability data include the test result for physical, chemical, biological and the microbiological parameters, and the product specific test like performance test. For example, it is a dissolution test for solid formulations. The variation in the stability data is a key consideration for extrapolation, statistical evaluation of the data, and assigning the shelf life. So these terminologies are very important to consider, as these terminologies and these techniques are used for shelf life estimation. The basic concept of stability data evaluation are single versus multi-factor studies and full design studies versus reduced design studies. So you may have heard bracketing and matrixing designs. Then the stability data to be presented in the tabular form and graphical form. Now consider you have completed the stability studies for the formulation batches, 3 number of batches, and now you have the data. For example, I will say you have a data of 12 months stability. So you have done accelerated stability study for 6 months, intermediate studies and long-term studies are ongoing, and now you have 12 months data. So based on that, the shelf life can be estimated and extrapolated. This extrapolation technique is used to know or to get the idea about the future data like 18 months data or 24 months data. So extrapolation in the simple meaning is that estimation of unknown value based on the existing value. If you know the data of 6 months, like here you take a plot take it as a impurity and here as a time. So this 6 month data you have, and you know the increase in the impurities from initial. For example, I am taking this, and you can extrapolate this for 12 months. So this is extrapolation. Or if you have 6 month and 12 months data here, then you can extrapolate up to 24 months. So based on the available data, by extrapolation you can know the future data. So the available data is used to predict the future data, and the data trend is also considered here. Always remember that the quantitative attributes are only considered in the extrapolation. Quantitative attributes are the assay or degradation products which are required to be extrapolated. The extrapolation of the data is required for long term and the accelerated conditions. You cannot extrapolate the physical attributes like appearance or description because these don't have the quantitative values. The variability in the data and significant change at accelerated condition is critical to be evaluated for assigning the shelf life. So it is very important to have the understanding about the stability data. Once you have the stability data, you look at that data thoroughly. You should have the release specifications and the stability specifications in your hand, and then you correlate that data and then do the extrapolation. If the data shows variability, like sometime assay is coming 97, sometime it is going 103, sometime again it is coming to 100, sometime it is going to 96, so the batch is same. Also you should consider why variation might be there. Root cause analysis should be done. If it is an analytical error, then if required, data can be generated once again by testing or repeat testing for your understanding and performing the extrapolation. But if it is variable, then that variability should be considered. Now if you have the data of stability study, so in general I have divided these stability data evaluation and shelf life estimation by two options. So option 1 is shelf life estimation based on long term stability data. If the accelerated stability data is within the acceptance criteria and no significant change is there. And option 2 is shelf life estimation based on the intermediate term stability data if accelerated stability data is not within the acceptance criteria or shows variability with significant change. So first terminology is significant change. Significant change means change in any of the quality attribute beyond the specification range. And as in the shelf life estimation, we are going to consider the quantitative parameters. So here the significant change will be change in the potency by 5%, change in the assay by 5%, and change in the degradation products or related substances or the organic impurities out of the specification or not meeting to the specification. So option 1 is when the accelerated data is okay and no significant change is there, variability is not there. And second option is there where the significant change has occurred at accelerated condition, but the intermediate condition is okay. Now see how the expiry period is assigned. So first condition, no significant change at accelerated condition. And if you have accelerated stability data of 6 months and for example long term stability data for 9 months. So as per the guidelines, it is given that shelf life can be up to twice but not more than 12 months beyond the period covered by the long term data. Now you have accelerated data 6 months and long term data is 9 months. So simply you will have 12 months as a shelf life. If you have 12 month long term data, twice of the 12 month it will become 24 months. If you have long term data of 18 months, so 18 plus 12 will become 30 months, because here it can be up to twice but should not be more than 12 months beyond. So if you twice this 18 months, it will go 36 months, and it cannot be twice, it cannot be more than the 12 months beyond. So it cannot be twice of the 18. Now it is 12 plus 18 is equal to 30 months. If you have 24 months data, then you can get 36-month shelf life. And if you have 36-month data, then you cannot go beyond that. 36 months will be the maximum shelf life. This is as per the option 1, where accelerated stability data is okay, there is no significant change. Now we will see the another option where significant change is there at accelerated condition but no significant change at the intermediate condition. So here I have considered intermediate and long term data. So if you have 9 months data for long term and the intermediate condition, then there are a requirement of data amenable to statistical analysis or not. So what is the meaning of this data amenable to statistical analysis? These terminologies are very important to be understood. So see, amenable to statistical analysis means there is the data which can be taken for statistical studies or statistical analysis, and data is not much variable. Data doesn't have outliers. In simple meaning, data don't have outlier. Data is inline and variation is not there. If data is not amenable to statistical analysis, then different type of shelf life period is given. So if data is not amenable to analysis, shelf life can be up to 3 months beyond the period covered by the long term data. And now if you have 9 months data, then only 3 months you can extend it, and it will become 9 plus 3, it will become 12 months. If you have 9 months data but data is amenable to statistical analysis, that means data is not having any variation, and you can take that data for statistical analysis, then shelf life can be up to 1.5 times but should not be more than 6 months beyond the period covered by the long term data. So 9 months will become 13.5 months and 12 months will become 18 months. So here in 18 months, it is covering both the criteria, like 12 months plus 6 months, it is becoming 18 months. Now if the product is not stable at intermediate condition also, some of the products are very sensitive, and these products are sensitive to temperature. That's why these are not stable at accelerated condition of 40 degrees Celsius, and intermediate condition of 30 degrees Celsius. But these formulations are stable at long term condition. So if there is a significant change at intermediate condition, for example, 12 months intermediate condition is showing significant change, but the long term data shows no significant change, then shelf life should not extend beyond the period covered by the long term data. In addition, a retest period or shelf life period shorter than the period covered by long term data can be assigned. So for those products, the stability data is okay only for the long term condition. That's why the shelf life cannot be extended, and shelf life will be equal to the long term data only. If you have 12 months data, you will get 12 months stability period. If you have 24 months data for long term condition, you will get shelf life of 24 months. So from this we can understand that if product is stable at accelerated, intermediate and long term, then you will get more shelf life or longer shelf life. If it is not stable at accelerated and intermediate, then shelf life can be assigned only based on the long term stability data. Now here I have taken the example from EMA guideline, and just for understanding I have taken this. Shelf life estimation with upper and lower acceptance criteria based on assay at 25 degree and 60% RH. So this is the example of extrapolation. Here assay is given, percentage of label claim. Here time points in month are given. Now you consider the lower and upper specification for assay as from 95 to 105%. If your initial value is 100 and the product is kept at stability condition of long term, then in how much time it is coming down to 95%. So by figure you can see here that it might be around 36 months, or it might be here with the lower confidence limit of 27 months. So this is the example of extrapolation for assay. Here the data is available for 12 months, and based on that the final time is extrapolated at which the product will have the lowest potency or will have the significant change with change in the 5% assay of the label claim. Now another example is estimation with upper acceptance criteria based on the degradation product at 25 degree and 60% RH. So here degradation product percentage is given, here time point in months is given. The degradation product is 0.5% at initial and the limit is 1.5%. So as a upper confidence limit, it is touching here at 30 months. So this is the example of extrapolation. You have the 12-month data, and based on that trend you are going to extrapolate. Now stability data evaluation and shelf life estimation generalized statements are given. The shelf life of the product is approved by the regulatory authorities after complete review of the application, but the manufacturer can give the tentative shelf life to continue the stability study and to perform the bioequivalence studies, or to have the understanding about the stability storage requirements, and also to continue the stability studies the tentative shelf life is required. The 24 months long-term stability data is required for approval of the 24 months as a shelf life or expiry period. So generally, the regulatory authorities approve the product after having a stability data of 24 months. Shelf life extension can be done and requires the stability data. If shelf life more than 36 months is required to be proposed, then the regulatory agency requires the data. And after getting that long-term stability data only, the regulatory bodies like FDA, EMEA, ANVISA and other country-specific regulatory authorities will approve the product with the final shelf life. So this is regarding the shelf life estimation and stability data evaluation. The reference I have taken mainly from this evaluation of stability data, CPMP SCH guideline. Also I have studied the US FDA requirements, and based on that I have made this video. As the formulation is required to be stable, for generic formulations the stability should be equivalent or better than the reference product. So always we have to understand about the stability requirements. Thank you for watching the video. Kindly do like, share and subscribe to Pharma Learning in Depth channel. Thank you.